Starting. All attendees are in listen-only mode.
Yes, hi everyone. My name is Shellen McCoy, and on behalf of the Foundation for Conservation of Atlantic Salmon, I just want to say welcome today to today's webinar. If you wish to have simultaneous translation, you want to hear the presentation in French, you can go in our chat and you'll see the instructions there for you to be able to have that. But for today, I'd like to welcome Robert Lennox. Thank you for joining us today. Just to tell you a little bit about Robert, he's a scientific director of Ocean Tracking Network. He completed his undergraduate degree in marine biology at Dalhousie University before moving to his PhD studies in Carleton University. Following his PhD, Robert was hired as a senior researcher at NORSE, Norwegian Research Center, where he established a research program in Bergen focused on fjord ecology, which included the establishment of acoustic tracking array to study Atlantic salmon migrations and the impacts of hydropower. Also the effects of disease on sea-run brown trout, urban ecology of Atlantic cod and European lobster during a restoration, and behavior of spiny dogfish. During this time, he supported the establishment of the European Tracking Network via Horizon Europe partnership grants. At Dalhousie, the Lennox lab uses acoustic tracking systems to study Atlantic salmon and brook trout, as well as the ecology of the critically endangered Atlantic whitefish and the migrations of Atlantic mackerel. All of Robert's research revolves around better understanding the link between animal movement and the environment with a slant towards conservation. As a scientific director of OTN, Robert is supporting scientific advancement and innovation in the global aquatic animal tracking community by supporting equipment loans and optimal use of the infrastructure. So to no further ado, here's Robert who will be speaking on tracking salmon use of lake habitats using acoustic telemetry. So Robert, the floor is yours, and of course at the end we'll allow time for questions and answers. So welcome.
Amazing. Thank you so much for that kind introduction. I cannot see anybody that is here, so I don't know how many people are here, but welcome and thank you for joining. I asked Charlene how many people, or if anybody was going to show up, and she said that yes, probably. So that's very exciting. I really like to have the opportunity to share all this with you, and I've been to some of these seminars before. I got to see Ian Bradbury's presentation for FCAS last year, and I thought, wow, maybe someday I will get invited to do something like that. That's super cool, and now I get to do it. That's awesome. And when I got the invitation, I planned to make a presentation really focused on kelp migrations and sort of a more comprehensive look at some of the work that we've done on salmon kelps. But some of the best parts of that research still isn't ready to present. So it would have been kind of a short presentation. And so I've pivoted to a much bigger, longer story with lots of data about the tracking salmon in lake habitats that I did in Norway in a previous life. And the way that a lot of these tracking studies work, it takes some time, years and years in some cases, to really compile all the data, get them analyzed, and be able to tell the story. So this is a look back on kind of the last half decade of my life, a project that's taken up a lot of time and I've been really passionate about and really enjoyed doing. When I was a senior researcher with NORSE, this project was funded by the Research Council of Norway. And in my current role at Dalhousie and with Ocean Tracking Network, I've continued to follow up on some of the data as they continue to roll in across the years. So it's been a cool project. It was, and I'm going to tell you all about it. So I don't need to introduce myself because Charlene already did. That was very nice. I'm scientific director here at Ocean Tracking Network. I'm also an associate professor at Dalhousie. Grew up in Ottawa, fascinated by fish, came to study marine biology, and I never stopped after that. There's a nice salmon that working on in Norway and in one of the rivers in Sunnmøre, which was super fun. So this story is going to focus mostly on the Vaso salmon, which is a really amazing population of salmon in western Norway. The Vaso River is at the end of a long fjord, and it's a steep river, but it's a river with two large lakes, which makes it quite interesting. And looking through the literature on lakes in salmon rivers in Norway, at least, we found that actually about 30% of the rivers there have lakes in them. So what salmon are doing in lakes and the ecology of salmon in lakes is actually kind of an interesting and important question. And the Vaso salmon is fairly well known in Norway as being an extremely large-bodied specimen. So decades ago, they were some of the largest salmon in Norway, and it was an extremely productive run for many, many years. Unfortunately, like many other rivers that we know of nowadays, the population started to collapse. And now, since the 1980s, the population's been in a low state due to a variety of factors. And Vaso's a river with a lot of human impact. So there's some channelization of the river to accommodate roads and farms. There's bridges built across the river. There's farmland on the side that probably contributes some excess nutrients. There's also some historic acidification effects. It was heavily fished, so you can see these gentlemen on the right catching some very large fish, and some of the numbers of salmon that they caught in Vaso in the mid-century are staggering, not only using recreational fishing, but using a variety of fish for subsistence fishing. And then there's also the impacts of the aquaculture. So there's a lot of escaped farmed salmon entering the river and introgressing their genes with the wild salmon, and then the juvenile salmon, the smolts that are migrating out of the river, they also encounter quite a bit of pressure from sea lice in the fjord. So it's a river with a lot of impacts, and it's not an easy place to point a finger at exactly what has happened in Vaso. But it's such an iconic population and such an important one to the local community at a national scale. It receives a lot of attention, and there's a lot of interest in trying to figure out exactly what could be done to get this population back on track. And I guess I'll rewind a bit as well and say that one of the local concerns in Vaso, and I'll show a little bit about this later, is that there is a power station that was built in the river, and it's not a dam the way that we have in Canada. We don't have a lot of these. It's a high head power station that takes water from the mountains and tunnels it down from the mountains into one of the lakes. And so that the water is coming from the hypolimnion in the lake. So in the summer it's colder than the ambient water, and in the winter it's a little bit warmer. And there's concerns that the changes to circulation and temperature may be affecting the smolt migrations. So we started to look into this with the industry partners and with the local management, the environmental directorate in Norway, and we started to try to compile what we knew about salmon in lakes. I drew this conceptual figure myself, and we wrote up a paper, a review paper that's published now in Canadian Journal of Fisheries and Aquatic Sciences, focused on the ways that anadromous salmonids use lake habitats to complete their life history. At the time, there was a paper that had recently come out, I think in Fish and Fisheries, about salmonids in lakes, but it was really focused on resident fish, not on anadromous fish. And I think, if I recall correctly, that paper only had a few mentions of anadromous fishes. So this paper really focused on how migrating salmon, trout, steelhead, Arctic char use lake habitats, and what the knowledge is about how the parr are using it, how the smolts are migrating through, how the adults use it before or during spawning, and how the kelts use it after. I hope this works so badly. But this is the power station in the lake that is in question. So it's just a short clip, but you can see that that's a tunnel, and you can see up here, maybe I should put on my laser pointer, I love that. You can see up here that didn't work, yeah. You can see up here the cliff, the mountains, so that's sort of on top of there's a plateau where the mountain lakes are. A series of complex tunnels bring the water down through this through a turbine, and then it gets discharged through this tunnel into the lake. And just one more time for anybody that missed it, it's not a massive flow, certainly not compared to the flow from the river, but that water tends to be colder than ambient through most of the time that the salmon are in the lake. This is from Cecilia Nilsson's thesis as well. So I'm going to talk about two components of this project. The project ended up getting funded by the Norwegian Research Council and, with collaboration from two hydropower companies to better understand how these high-head power stations influence the ways that salmon and sea-run brown trout are using lake habitats. And we tagged both salmon and sea trout, and we tagged two different life stages. So we tagged smolts and we tagged adults. And we instrumented two different river systems. I'm going to talk mostly about Vaso, but we instrumented the two different river systems with receivers to track their movements up and down the system. So we had rivers in the fjord, we had rivers in Bolsta Elvi, we had rivers in Evanger, we had receivers in Bolsta, Evanger, Vaso, Vangs, and Stranda. So we were able to track all the migration up and down the system. So I'm going to start with some information about the smolt migrations through the lakes because that's one of the most interesting. This is going to be somewhat incomplete because even though it's been 4 years since we started this, there's still more coming. So Cecilia Nilsson, this is her PhD project, and some of the results are going to be work that she's done. There's also a hatchery on Vaso, I should mention as well. In Norway, they have gene bank hatcheries where they very carefully collect genetic material and then intentionally cross certain individuals based on their genetic phenotype to avoid inbreeding and negative impacts associated with that as best they can. So the hatchery's up at the top here. Most of the salmon that we tagged were in some way of hatchery origin, but we did a mix of hatchery and wild fish, and we focused on wild fish as much as possible. This is a look at the river in the springtime, a little bit early in the spring before the flood comes, nice and calm. It gets a little bit crazy, and then we're going to be tracking them moving down through the system in these different areas with a big focus on this first basin of the lake. Later on, I'll talk about the adults that get tagged at the opposite end. So one of the first things that we did as part of this project was evaluate the impacts of the tags on smolt migration timing, because we were concerned about selecting for early migrating salmon by fishing at a specific time. So we electrofished salmon smolts in order to tag them. And of course, when you electrofish in late March, early April, you have to discard a lot of fish that are too small to tag. Well, it turns out that those fish will migrate as well, but they just need to grow a little bit more. So I'm not showing the results of that study today, but we actually found that those small fish that are too small to tag early in the season probably could be tagged later in the season, and they end up migrating. So it's really, what we learned is you have to be really careful when you conduct these studies to get a representative sample of the population so that you're not biased towards those faster growing, potentially older fish that are larger earlier and migrate earlier. So that's something to keep in mind here as well. But we found that comparing sham acoustic transmitters and dummy acoustic transmitters with pit tags, which are obviously smaller, resulted in not major differences in the timing of recapture. We did this in a small river nearby also that has a wolf trap that cuts off the river completely, so we could recapture everything. But we found that the size of the fish really determined the time to be recaptured again. So these small fish that could only be tagged with these pit tags, they took the longest to be recaptured because they needed to grow long. They needed to keep growing and wait until they migrated. So, and you can see that the change in length within 30 days, some of those fish were growing up to 15 millimeters. So over a centimeter in just a month. So this late growth can actually be quite important and can determine whether or not you're biasing your sample based on the timing of migration. This is sort of a note because it's not something that's well integrated into our research here, but lesson learned that one of the things that we should be doing is making sure that we're tagging at multiple time points throughout the migration, which is important. So how do you track the passage of smolt around a power station with acoustic telemetry? We put out, in the first year, we did this 3 or 4 times. In the first year, we put out this grid array, and we were hoping that we would be able to position the fish using YAPS or some sort of positioning solver to get a nice look at how they move through. You can see my red laser pointer, hopefully, and you get a nice look at them moving around the bend. We did some range testing with several tags in this really important area, and we found, okay, you know, the polygons of detection, 95% polygons around the detection sort of seem to overlap. So we felt pretty good that we were going to have overlapping detections and it would be easy to position the fish. That turned out to be not the case, at least immediately off the bat. Just a reminder that Vosso is coming in here and the smolts are having to migrate past the power station that's approximately here. Right, yeah. So this is Erland Hansen. He's now doing a PhD with us on a project about otter predation in Norway. But for his master's, he was the first master's student that I ever supervised, which is kind of fun. And we set up the first array for his study and tagged kind of a pilot project of smolts to look at how they move through the lake. And we used the Innovacy predation tags, the V5s on 180 kilohertz. We didn't get a ton of data, but one thing that we wanted to figure out was, or one thing that we realized was that the smolts were not moving quickly through this lake. They seemed to spend quite a bit of time in this first area, and turning that corner seems to be quite a challenge. And you could easily sort of wonder whether or not that has to do with the power station. You have this collinearity problem though, with the power station being right at this tricky turn. So Erland ran some simulations. He calculated the most likely step lengths and turning angles that the smolts would have, and he ran simulations to see how long it would take a smolt to reach the end of the lake if it was just moving according to random principles with a realistic step length and turning angle. And he found he was able to actually approximate how quickly the smolts in the real smolts got out of the lake with these simulations, which suggested that they weren't really using directed movement very much. And that kind of makes sense because we think that the smolt migration is somewhat rheotactic and they follow the flow. And so they're following the river down Vosso, and then they get into this first basin of the lake, and all of a sudden, unless they're timing it perfectly with the big flood, they're getting into a lake with no rheotactic cues. It's just a completely still, calm lake with no cues. And so they start to probably get confused, move around randomly, wait for the flow to come up. But what we found from these predation tags as well is not only are they spending quite a bit of time in this lake, but they're also getting eaten in this lake at pretty high rates. And Erland caught this giant trout in the lake when he was working there, and it was kind of a signal of what was likely going on there. So that was the pilot. We weren't able to get positioning to work in that pilot, but eventually we were able to get it to work. We set out receivers in a pretty tight grid around the area of influence. So the power station comes in approximately here. We put a really dense array of receivers for Cecilia's PhD thesis, and we used YAPS, which is yet another positioning solver developed by Henrik Backtoft, which allows you to calculate the precise or most likely positions. You can see that there's still some error. Obviously, the fish did not fly over land in order to get to the other side of the lake. But for the most part, we got pretty good information about the detections, and they're cut off a little bit here by a bounding box that I implemented in YAPS. And you can see that the fish are swimming around quite a lot in this area. Again, you can see we didn't position very much here. One, because the flow is coming out of Vosso here, so it's quite noisy and hard to position. And two, because as we continued to fail to get positions, we started to creep our receivers in a tighter and tighter grid in hopes that it would work. Here's a nice picture of a salmon smolt and a nice little brown trout smolt. But you can see that they're using this whole area. They don't seem to be necessarily getting entrained anywhere, or but you can also see that they're not moving very quickly or directed through this bottleneck where the water's coming out. So we came up with something quite clever in our view, which was in response to a question from the management when we presented these preliminary results, which was they're concerned that the fish were actually getting thermal shock from the from the power station. So cold water coming down from the mountains, 1 to 2 degree difference potentially. And there was concern that, okay, this is a barrier, the fish can't make it past. So we actually put temperature transmitting sensors, temperature sensors into the transmitters so that it would send data to the receiver about what temperature those fish were encountering when they were in the lake. Oh, yeah, that's out of order for some reason. So I was able to match the positions of some of the smolts that had these temperature sensors, because we didn't put them in all of them because of the size of the tags and the size of the smolts. They could only go in the big ones. And actually it turned out that the only data that we got from these tags was from some of the trout smolts. So keep in mind that these are not salmon tags. And you can see that there's some interesting patterns in the temperature. You can't see time here, so not obviously, but it certainly warms up as time goes on. So later in the season, we tagged and released most of these fish in late April, early May, and we're detecting them into June. So over the course of that month of May, it's getting brighter, the sun's coming higher, the river and the lake are warming up, and some of these warmer detections, 6-7 degrees, are probably from the second half of May, and some of the colder, more purple detections are probably from smolts early in the season. I ran that through a model to kind of predict the temperature experienced by a fish in different parts of the river. This is a generalized additive model with a spatial field as a two-way interaction. So an interaction between the X and Y axis and temperature as a predictor. And you can see that it gives some nice predictions of where the fish are likely to encounter different temperatures. And it's only predicting about a 1 degree difference in the experienced temperature. And we did this across time too. So we're getting different panels from the 20th of April to the end of April, starting into the middle of May, and then the end of May. And you can see that warming up predicted by the model. And you can start to see as well that there's a little bit of a signal in terms of this area immediately adjacent to the power station having a little bit of colder water signatures. So a bit warmer here, 5 to 6 degrees, whereas a bit colder here, 4 to 5 degrees. And that was kind of a signal that, yeah, there is a thermal effect. We knew that from measuring the water temperature. That cold water sinks pretty fast because it's heavier than the lake water. So it comes out of the power station, floats on the surface for a little bit, and then sinks down to the bottom. So it's not surprising that the whole lake is not apparently affected by this cold water discharge. The next step to this was to take some salmon smolts and put them into a respirometry chamber. So we did kind of a cold shock experiment in the lab that I don't have any slides on, so I'm just going to describe it quickly because it's probably a question that you have. Is it a big deal for a smolt to go from 5 to 4 degrees or from 4 to 3 degrees? Well, we put a bunch of smolts in respirometry chambers at different temperatures, and we're going to have some results on that soon, hopefully, to figure out exactly how much what the metabolic cost of this kind of temperature change is. And it's a really nice design, I think, because it pairs the telemetry, the actual specific measurements of what that smolt is experiencing, because you measured it on board the animal. You're able to take those exact temperatures and test those exact temperatures in the lab using respirometry. I really like that design, and I wish I had more results to present on it. So I mentioned the predation aspect. The other thing that we did with these smolts was we integrated some depth sensors. This was early on in the study when we were still trying to get a handle on what was going on in the lake, and we weren't really sure how the smolts were moving vertically. And one of the concerns was that this lake is actually deeper than a lot of the North Sea, which is crazy. Most of the North Sea is like 100